Hello, everybody. Good morning and good afternoon, and welcome to today's webinar titled From Myelin Damage to Immune Dysfunction, Unraveling the Aging Brain. I am Sydney Panilla of LabRoots, and I will be your moderator for today's event. Today's web seminar is presented by LabRoots and brought to you by VizGen. To learn more, visit vizgen.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. You can also use the Ask a Question box to let us know if you happen to have any trouble seeing or hearing the presentation. Okay, I would now like to welcome our speaker for today's webinar, Dr. János Groh, Technical University of Munich, Institute of Neuronal Cell Biology. János Groh is a lecturer in experimental neurology at the medical faculty of the Julius Maximilians University of Würzburg and a senior research associate at the Institute of Neuronal Cell Biology at the Technical University of Munich. He earned his PhD in biology from the University of Würzburg in 2013. In 2023, he joined the Technical University of Munich as a group leader and principal investigator. His research focuses on inflammatory processes triggered by the dysfunction of myelinating glial cells in the nervous system. Using murine models and patient-derived human samples, he seeks to deepen the understanding of how interactions between neural and immune cells contribute to structural and functional changes in disease and aging. Okay, Dr. Groh, I will go ahead and hand it over to you. You may now begin your presentation.
Okay, so thank you for this kind invitation to share some of our work on aging white matter. So as a start, I want to remind you that vertebrate neurons are compartmentalized, and in many cases, their axons are ensheathed by myelin. Myelin is formed by oligodendrocytes and Schwann cells, which wrap around axons multiple times to form this lipid-rich and tightly compacted membrane structure. In the white matter of the central nervous system, myelin is formed with a differentiation of oligodendrocyte precursor cells into oligodendrocytes, and it allows for a saltatory conduction, modulation of signal transmission, and axonal support. So it is very clear that myelinated axons are really important components of higher CNS function. However, because of their unique properties and also their complex architecture, oligodendrocytes and myelinated axons are especially vulnerable to perturbation, and they are affected early in many neurological disorders, famously including those characterized by autoimmunity like multiple sclerosis. Importantly, previous studies have exemplified that primary defects in myelinating oligodendrocytes can induce neuroinflammation of high impact for white matter integrity and brain function. The lab of Rudolf Martini in Würzburg has shown this in a series of studies on a transgenic mouse model of Pelizaeus-Merzbacher disease, an inherited leukodystrophy that overexpresses proteolipid protein, the major myelin protein in the central nervous system. And here the group could demonstrate that especially activated microglia and cytotoxic CD8 positive T cells contribute to the chronic demyelinating phenotype and the axonal damage observed in this mouse model. Together with Rudolf Martini, we have subsequently also demonstrated this in a newly generated mouse model carrying PLP gene defects previously identified in few patients initially actually diagnosed with multiple sclerosis. And also in this model, we observed chronic inflammation in the white matter. So there is pro-inflammatory microglial activation, as seen in this labeling of the myeloid marker CD11B, and there is an accumulation of mostly CD8 positive cytotoxic T lymphocytes in PLP mutants. Here shown in red, associated with damaged axons, showing dephosphorylation of neurofilaments and swelling in green. Such CD8 positive T cells normally recognize and fight infected or malignant cells. However, in case of inflammatory diseases like MS, they can also do unwanted damage in the nervous system. So for our experiments, we routinely assess the retinotactile system in mice. This system comprises distinct and very well characterized and also accessible compartments of the CNS, with the optic nerve representing a pure white matter tract. The retina contains the corresponding neuronal cell bodies and dendrites of retinal ganglion cells. This model system also allows non-invasive longitudinal monitoring of disease progression or therapy success. So we established optical coherence tomography, or OCT, in living mice to reduce animal numbers for research and also track retinal thinning longitudinally in the same animals. And indeed, we found with various genetic and also pharmacological approaches that immune deficiency or specific immunomodulatory treatment approaches can attenuate neuroinflammation, neurodegeneration, retinal thinning, as seen with OCT, and disease severity in these PLP mutant mice. So this confirmed that primary defects in myelinating glial cells can result in secondary inflammation that drives neurodegeneration. So why are we interested in secondary processes in models of relatively rare genetically caused diseases? We always felt that it is very important to also study such diseases and that they offer great opportunities to learn for more frequent diseases or conditions, for example, those associated with myelin defects and chronic neuroinflammation like MS. Moreover, we think that they are relevant for even more frequent conditions such as the normal aging process. Alan Peters, a pioneering electron microscopist, has shown decades ago that there are effects of the normal aging process on myelin and axons, for example, by the formation of such organelle-filled axonal spheroids. And in the last few years, there's growing evidence that also in aging-related neurodegenerative diseases like Alzheimer's disease, Parkinson's disease, and ALS, oligodendrocyte dysfunction, myelin alterations, and white matter inflammation play an important role in the pathogenesis. So underlining our observations in models of rare monogenetic disorders, we could recently show that neuroinflammatory reactions also drive degenerative processes in the central nervous system of mice during normal aging. Again, we identified a critical and previously underestimated role of CD8 positive T cells that damage myelinated axons, here again stained in green with non-phosphorylated neurofilaments. And this again emphasizes the vulnerability of myelinated fiber tracts. If we look at REC1 deficient aged, so if I say aged, I mean 24-month-old mice, in this case REC1 deficient mice, which lack functional adaptive immune cells, we observed less axon degeneration and neuron loss than in wild-type mice. And also in aged human frontal white matter without disease, we detected an accumulation of CD8 positive T cells and axonal damage. Similar to our myelin disease models, we observed that age-related inflammation and axon damage was not confined to the retinotactile system, but it also occurred in other CNS white matter regions, such as the cerebellar white matter and the corpus callosum, so the main white matter tract of the brain. Accordingly, this REC1 deficiency in aged mice also resulted in an improvement of rotor rod performance, so better motor coordination, and also prevented some aging-related cognitive decline as measured with a novel object recognition task. So we therefore think that the optic nerve is a very good model system to study white matter aging and disease. Moreover, in collaboration with Wolfgang Kastenmüller and Emanuel Saliba in Würzburg, we could show using FACS sorting and single-cell transcriptomics that these CD8 positive T cells resemble tissue-resident memory T cells, that they are heterogeneous, so there are several subpopulations, and that they show aging-related transcriptional changes. We then used bone marrow transfer experiments, which revealed that specific subsets of activated CD8 positive T cells damage axons by cognate T cell receptor dependent cytotoxic mechanisms involving the effector molecule granzyme B. And indeed, with novel technological developments, a detrimental role of CD8 positive T cells becomes increasingly recognized in many CNS disorders in which they have long been neglected, I would say. Not only in chronic autoimmune diseases, but also in normal aging and aging-related neurodegenerative diseases like Alzheimer's disease, Parkinson's disease, and ALS, as you can see in this list of selected studies. And together with Mika Simons and Özgün Gökçe, we could recently corroborate a detrimental impact of these CD8 positive T cells in aging white matter and show that they contribute to pathological interferon-dependent reactions in aging oligodendrocytes. Many of these studies also implicated interactions between microglia and CD8 positive T cells as a pathogenic aspect, similar to our findings in rare models with myelin gene defects. Mika and I recently summarized some of these findings on white matter aging and its impact on brain function if you want to go into more details on this. And due to these findings, we postulate that also in aging and aging-related disease, myelin perturbation can induce neuroinflammation that contributes to damage and degeneration of myelinated axons. So we are now further clarifying different aspects of myelin-related neuroinflammation in the diseased and aged CNS with the aim to identify feasible strategies to modulate this. And these aspects comprise three main research questions. So how does persistent myelin perturbation induce and maintain chronic neuroinflammation? How do these immune cells and neural cells interact to promote axon degeneration? And finally, what are the mechanisms within the aged and diseased CNS that allow resilience and functional preservation despite ongoing pathological alterations? Microglial cells are related to several of these questions, since they are known to be essential orchestrators of neuroinflammation, but they also have important properties in reparative and protective responses in neurological diseases and in aging. So it is long known that white matter compartments show prominent changes during aging, and that these affect microglial cells, as shown in seminal studies by Wolfgang Streit, for example. A more recent work by Mika Simons' group has further illuminated and also related some of these processes to protective functions of microglia. We have recently shown in our mice with distinct PLP defects that microglial phagocytosis of abnormal myelin can be protective against CD8 positive T cell driven axon degeneration. So we therefore now focused on our favorite white matter compartment, the optic nerve, to characterize myelin and microglia in aging. And when we looked at optic nerves from aged mice, there was an increased frequency of fibers with redundant or fragmented myelin, so abnormal myelin. Moreover, there is a slight increase in the number of non-myelinated or thinly myelinated axons, and there is a fingerprint of microglial myelin phagocytosis indicated by an accumulation of myelin fragments and lysosomal storage material within microglia. We also detected a continuous increase in the number of microglia which cluster in nodules and show changes in cell morphology, so they become more circular. Moreover, there is an increased expression of the activation marker CD11C, interestingly showing a steep increase in expression between 18 and 24 months of age, fitting to what we observed for the accumulation of these CD8 positive T cells. We also detected interactions of activated microglia with CD8 positive T cells in the white matter of aging mice. So to address the function of microglia in aged white matter, we used two different approaches. We used pharmacological depletion using the small molecule PLX5622 at 300 milligram per kilogram chow, which inhibits the colony-stimulating factor 1 receptor signaling and results in dose-dependent microglial apoptosis. And we investigated aged mice lacking the fractalkine receptor CX3CR1, which is important for protective microglial functions. So these mice have microglia displaying a more pro-inflammatory phenotype with impaired phagocytosis. So we first quantified white matter microglia and their activation in both groups in comparison to untreated adult and aged mice. And interestingly, PLX treatment only partially depleted microglia in aged mice, while CX3CR1 deficiency did not lead to significant changes in number. However, when looking at their activation using CD11C as a marker, both approaches showed that there is increased pro-inflammatory activation of the remaining or CX3CR1 deficient microglia in aged nerves. When we compare microglial morphology in these PLX-treated aged mice, we observed that there were large regions in the nerve lacking microglia. And some of these microglia that remained after PLX treatment showed increased circularity and buildup of autofluorescent storage material resembling lipofuscin. And this was also reflected in ultrastructural analysis. In regions likely devoid of microglia, we found these complicated, redundant myelin figures, which I like to call the myelin maze. So you can try to follow these really complicated windings of individual myelin protrusions. And when you find microglia, they are frequently completely full of myelin remnants and lysosomal storage material. So they seem to try to compensate the loss of their partners and become even more dysfunctional and burdened with poorly digestible myelin. Quantification then showed that in both cases of interfering with microglial function, there are more fibers surrounded by redundant or fragmented myelin and less thinly or non-myelinated fibers, indicating impaired phagocytosis of aberrant myelin. Here you can see that there really are a lot of these strange myelin formations also in the CX3CR1 deficient mice, indicating that this is usually removed by microglia during the aging process. So the partial depletion or CX3CR1 deficiency enhances aging-related microglial activation and dysfunction. To better characterize microglia after PLX treatment in aged mice, we performed single-cell RNA sequencing of FACS-enriched microglia with fixed RNA profiling to avoid artifacts. Single-cell RNA sequencing of microglia from aged compared with adult mice demonstrated the presence of several white matter-associated microglia clusters that typically arise in disease and aging. So I will not go into too much detail, but we found that these activated microglia clusters shared the downregulation of homeostatic markers and the upregulation of activation markers, reminiscent of the disease-associated microglia program. The presence of multiple of these activated microglia states might explain their diverse roles in aging white matter. We found that the CSF1 receptor inhibition more efficiently depleted homeostatic microglia, and that the activated microglia are more resistant and survive the treatment. Moreover, we confirmed that the remaining microglia after PLX treatment in aged mice showed stronger expression of these WAM marker genes, indicating more pro-inflammatory activation, leukocyte stimulation, lysosomal dysfunction, and senescence, confirming that the partial depletion of microglia enhances aging-related microglial activation and dysfunction. And of course, we looked for a link to CD8 positive T cell recruitment in the white matter. Importantly, both the partial microglia depletion and also the CX3CR1 deficiency resulted in a significant and specific increase in the number of CD8 positive T cells in optic nerves. And when we quantified formation of axonal spheroids by immunohistochemistry and the number of total axons by electron microscopy, we detected increased spheroid formation and an aggravated loss of optic nerve axons. And this also resulted in aggravated loss of RBPMS positive retinal ganglion cells in the retina of aged mice. And this exaggerated neural damage caused a decline in inner retinal thickness as measured with optical coherence tomography. Moreover, optogram analysis of visual function revealed a decrease in visual acuity in the treated or CX3CR1 deficient mice. So there's a really good correlation between increased microglial activation and dysfunction, CD8 positive T cell recruitment, aggravated axon degeneration, and behavioral decline in aging. Single-cell transcriptomics of T cells also indicated that PLX treatment reduced the expression of inhibitory marker genes and increased the expression of cytotoxic effector molecules. We confirmed the increased expression of the specific cytotoxic molecule granzyme B in CD8 positive T cell granules using super resolution expansion microscopy actually. And we saw that the CD8 positive T cells more often contacted CD11C positive activated microglia after PLX treatment. And importantly, when we depleted microglia in aged CD8 knockout mice, we did not see this aggravation of neurodegeneration. So these findings indicate a protective role of some microglial states in aged white matter, which is supported by several recent studies. However, they also indicate that maladaptive microglia activation promotes chronic pathogenic neuroinflammation by CD8 positive T cells during white matter aging. And this led us to another important open question. How do these activated microglia contribute to the recruitment of CD8 positive T cells into the white matter parenchyma? To better understand this, we performed Murfish-based single-cell resolution spatial transcriptomics of optic nerves using a custom-designed 500 gene panel. And here you can nicely see that the MERScope system helped us to accurately identify the major cell types in adult, aged, and also PLX-treated aged nerves in a relatively unbiased manner. And in summary, Murfish confirmed our previous observations that partial microglia depletion enhances aging-related microglial activation and dysfunction. We then used the ability of Murfish and the MERScope platform to study spatial expression profiles in cell neighborhoods at subcellular transcript resolution, as you can see in this nice example of a longitudinally cut optic nerve from an aged mouse. And we observed that the T cells showed a significant neighborhood score for CXCL10 expressing cells, which we mostly identified as reactive astrocytes. So this immediately jumped at us because CXCL10 is one of the most important chemokines for T cell recruitment, and we found it upregulated in our transcriptomic profiling of aged white matter, particularly after PLX treatment. So we then confirmed the increased expression of CXCL10 by reactive astrocytes using immunofluorescence and observed increased reactivity in PLX-treated mice. Microglia themselves also expressed CXCL10, but less frequently than astrocytes. We also validated that activated microglia, CXCL10 expressing astrocytes, and CD8 positive T cells often closely interacted in aged white matter. Our single-cell profiling also included oligodendrocytes and astrocytes in addition to microglia, and these glial cells also showed aging-associated reactivity. Computational prediction of cell-cell interactions using CellChat indicated complex interactions between different glial cells and also between reactive glia and T cells. And this analysis again highlighted secretion of CXCL10 by reactive microglia and also by astrocytes, which acts on the CXCR3 receptor expressed by T cells. It also indicated that microglial activation during white matter aging triggers this response of astrocytes and their induction of CXCL10 expression. We actually confirmed this in TREM2 deficient aged mice, which locked microglia in a homeostatic state and decreased CXCL10 expression. So we concluded that the recruitment of these CD8 positive T cells into the CNS of aged mice via activated microglia occurs via release of CXCL10. And we therefore looked at white matter of aged CXCL10 deficient mice. And indeed, we found that CXCL10 deficiency strongly attenuated the recruitment of CD8 positive T cells into the aged white matter and mitigated aging-related axonal spheroid formation and axon degeneration. To approach this CXCL10-mediated T-cell recruitment from the receptor side, we then devised a bone marrow transplantation strategy to knock out its receptor CXCL3 in adaptive immune cells. And this also resulted in decreased T-cell accumulation and axonal spheroid formation in white matter with increasing age. So this convinced us that CXCL10 CXCR3 signaling is involved in the recruitment and residency of CD8 positive T cells to aged white matter. Our combined results thus show that microglia activation orchestrates CXCL10 mediated CD8 positive T cell recruitment to promote aging-related white matter degeneration. So these findings have recently been published in Nature Neuroscience, and I'm especially happy that the artwork my 4-year-old son created, of course I had to help a little bit, but he did most of the work, was chosen for the cover of this issue. And I think it's a wonderful coincidence that our new study on aging concludes with the cover of a 4-year-old. So we represent different glial cells as paint splotches in aging white matter. We have maladaptive microglia and astrocyte states, which promote the recruitment of CD8 positive T cells, here shown in red, and contribute to the formation of abnormal myelin, here in purple, leading to axon degeneration and behavioral decline. So efficient myelin phagocytosis and turnover by microglia seems to counteract axon degeneration and can enable the resolution of inflammation, at least under specific conditions. And if this protective response fails, chronic inflammation drives the deterioration of structure and function. So in conclusion, chronic myelin-related neuroinflammation contributes to structural and functional decline of the CNS in disease and during normal aging. Modulating this inflammation might offer distinct therapeutic opportunities of high translational relevance for myelin and aging-related neurological diseases. And ideally, these might suppress detrimental neuroinflammation, but still allow or even foster resilience and regeneration. And we are convinced that novel high-resolution profiling technologies can enable the discovery of such modulatory approaches. Finally, as an example for this, and also a short teaser, I want to point you to our new paper, which actually was just published online in Immunity. Here we used the ability of the MERSCOPE platform to perform high-resolution spatial transcriptomics of chronic active MS lesions. And we found maladaptive interactions between inflammatory glial cells and CD8 positive T cells at lesion rim niches. And we uncovered translationally relevant strategies which might disrupt such pathogenic inflammatory interactions. And with this, I would like to thank the people that made these studies possible. Rudolf Martini from Würzburg, Mika Simons in Munich, and all present and previous group members and collaborators. Thanks again, Visgen, for the invitation, and thank you all for your attention. And I'm, of course, very happy to discuss your questions. Thank you very much.
All right, wonderful. Well, thank you again, Dr. Groh, for your very informative presentation. We will now go ahead and move into the live Q&A portion of the webinar. So if you happen to have a question that you would like to ask, please go ahead and do so now. Just type your question into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for. Okay, let's go ahead and jump in here. The first question that we have is a two-part question. And this one says, do you view axonal myelin damage as the initial trigger, or is vascular inflammatory change the instigator? And how might that influence potential therapeutic timing?
Okay, so thank you. This is a very interesting question, of course, that is a bit difficult to answer. So in aging, what comes first, right? This is much more clear in our myelin mutants, where we have a gene defect that affects oligodendrocytes. In aging, this is much more difficult to answer, but our hypothesis is that persistent oligodendrocyte dysfunction is probably something that is one of the instigators of this process. One of our results actually also points in this direction. If we block microglial reactivity with a TREM2 deficiency, this does not really ameliorate the abnormal myelin of oligodendrocytes in the white matter. So one hypothesis is that oligodendrocyte or myelin dysfunction is one of the initial triggers of this process. How this is linked to vascular changes is still pretty unclear, I would say. So we don't know exactly where all starts.
Okay, fantastic. The next one that we have here says, did you manage to detect T cells using MERFISH, and were they localized close to the microglia or any consistent cell type?
Yes, we detected T cells with a MERFISH. We found them in optic nerves, and we found them in specific niches, and around those T cells, we found mostly CXCL10. So this is basically how we came to focus on the CXCL10, CXCR3 signaling. We found that CXCL10 was mostly enriched in reactive astrocytes, but there were also activated microglia that expressed CXCL10. So, and then this is basically how we came to the idea to look into CXCL10 deficient aged mice.
Perfect. We have another here that says, you said you used optical coherence tomography, OCT, to reduce animal numbers in research. Are there other ways to reduce animal numbers, and have you used other animal models in research, such as fish?
So yes, we use optical coherence tomography as one non-invasive readout because it is related to one of the systems that we like to study, the retinotactile system, and that allows us to non-invasively track thinning of the retina. So I have not personally worked with fish. I mostly do research in using mouse models or human material, for example, but it can also, of course, help to reduce animal numbers using other model systems, so also specific in vitro systems.
Okay, great. The next question that we have here says, do you see regional vulnerability of white matter in aging? For example, anterior versus posterior tracts?
This is a good question. We see that specific domains in the white matter seem to be affected more strongly than other regions. Like, for example, if we look into the optic nerve, we see that many of these CD8 positive T cells accumulate in the more rostral part near the optic nerve head, where also the blood-brain barrier is not as tight. And we also see that there seems to be an rostrocaudal axis, so we see more inflammation in the more anterior parts, actually. Yes. And also in the corpus callosum, for example, there seem to be specific domains where we see a lot of inflammation, for example, above the ventricles. There's a lot of inflammation going on. So I would say, yes, there are specific regions where we see more changes.
Okay, great. It looks like we have one final question here, so we'll go ahead and wrap up with this one. And it says, how does spatial profiling help you with these studies?
So For us, it was clear that we need technology that allows the identification of relatively rare cell types like these T cells, as was also already asked in another question, because in the aging process, it's not a massive amount of these T cells that are infiltrating the brain. So we needed something with high resolution, and we wanted something that helped us to screen the microenvironment of these T cells for clues to figure out why they are recruited and also retained in the white matter. And Murfish was a technique that helped us to answer these questions due to the high resolution and also the good spatial, the ability to spatially profile these transcripts. Okay.
Okay, great. Well, thank you again, Dr. Groh, for your time today and for your very important research. We would also like to thank LabRoots and our sponsor, VizGen, for underwriting today's educational webinar. Before we go, I would like to thank the audience for their interesting questions. Questions that we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. As a reminder, this webinar can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. All right, until next time. Thanks so much, everybody. Goodbye.
Thanks. Bye.